that I wasn't going to post the old exams because I didn't want you to get distracted by questions that wouldn't be like the ones you'd see this year. But I picked a few for class today that are pretty representative of the kinds of things you're going to see on your final. So we're going to go through them. The podcast is recording, so that should be up this afternoon if you want to review them. But please go ahead and give them your best shot. I'm going to try something. See what happens if I do this. We'll vote again. Okay, so the question here is how many different mutant alleles will be present in the population of 50 offspring? All right, I'm going to stop it. You guys actually did something different than section A did. You guys got it right. At least in my opinion, it's right. So one of the props of this question is this was on an old exam and people did terribly. Most people actually picked 2, I think. So it's confusing. I think you guys understand the concept, but the idea is each diploid individual has how many alleles? 2. 2 alleles. Each diploid person or fly will have 2 alleles of every gene. How many are there in the population? In this case, 3. There's the mutant A, mutant B, and mutant C, and all the 50 offspring will have one of those 3. So there are 3 different, that's why I inserted this word, but it didn't change how you answer, maybe you didn't notice I did it. But different alleles. Some people might have thought 100, that was a popular answer in section A because there's 50 flies with 2 alleles. So in a way there's 100 alleles, but there's only 3 different alleles. Does anybody have any questions about this? Yep. Where does it say that it's recessive? It doesn't matter. So the question was, I think, for this one, how do you know it has 2 A alleles? No? Why is it mutant? Okay, so because the lab is working with 6 mutant alleles. So I understand your point. That's a reasonable point. So it could have been that this one had 1 wild type and 1 A. So questions like this are really tricky to write, and I may have changed the question a little bit from its original and messed that up. But that's a good question. Yes?
How can we know for sure that the mutant alleles GEFS.B and GEFS-C will be passed on to the offspring?
How can you know that they will be passed on?
If it's heterozygous,
Okay, so this fly 1 is homozygous. Oh no, so it's okay. So now I've got yours figured out too. So it's hard when I don't write the question. I know the answer, but fly 1 is homozygous for mutant A. Let's get rid of the whole guess. Okay, so it's AA. And then fly 2 is a heterozygous. It has 1 allele of B and 1 allele of C. So if you draw a Punnett square, you'd have AA, B, C. So you can get any combination. You can't get, so you can get A and B or A and C. For the, yes?
Fly 1 and fly 2 are mating.
Yes, fly 1 and fly 2 are mating. Producing offspring. Anything else? Yes? Okay, that's a good question. So two different mutant alleles were sometimes students get confused about, we talked about homozygous recessive mutants, and now this one has B and C, and they're both mutant. Well, remember in class I talked about cystic fibrosis, and I put a problem up there where one person had the delta 508 mutation, and another person had an entirely different mutation, and both of them mess up the cystic fibrosis transmembrane protein. So both of them are recessive mutants that will trigger CF, but they're different mutants. They have a different sequence. So maybe this one is a nonsense, this one's a missense, and this one's a frame shift, for example. You can mess up a protein many different ways. Does that answer your question? You don't look satisfied.
Like if I would write the A for homozygous being like two labels, if I would write the B for
Okay, so that's a good question. So how would you write this down? Because there's only one gene. So this gene causes the disease GEFS, so maybe I'd call it G, and then I'd put a little superscript A, and a superscript B, and a superscript C. Right, so it's GA, GB, GC. Like when I write the mutant X chromosome, I usually write X with a little m at the bottom, or you could write X with a little m at the top. When we did vestigial, it was vestigial plus is the wild type, and VG with no plus is the mutant. So it's one gene, so it gets one letter. Nice question. Yes?
For B and C, it signifies that those are mutant alleles.
Yes.
It signifies that that's a mutant allele.
I see. Well, so the question was, how do you know it is mutant? I probably think it would be better to say the GEFS-A mutant allele, but it does say I was heterozygous for two different mutant alleles, so different mutants from A. That's why it's so hard to write these questions so they're not confusing. And we agonize over it and go back and forth with the exams. Like 3 or 4 people look at all your exams. So we do our best, but there are problems. And you do have the option of talking to us about it afterwards if they're confusing. This problem is confusing. I did not put one, I haven't actually done your final yet. I will not put one exactly like this on your final, I promise, cross my heart, that the concepts could be in a question on your exam. So if you get the concepts, diploid individuals have 2, there could be 2 alleles in a population like this, there could be more alleles. So the maximum number of alleles possible in this case would be 4, because you're mating 2 flies. So they could have a maximum of 4 different alleles. Okay, thank you for all your questions. Those are helpful for me. All right, let's try this one. You guys didn't do so great on this in class. So draw a Punnett square, try and get it right. Okay, you guys are doing very well. Go ahead and finish up your squares if you have not. Okay, C is the right answer. Very good. So if you draw the Punnett square, it becomes pretty clear. Half of the daughters will not have Fabry, but they will be carriers. Right. Okay, try this. you got this right. I was hoping it would be all of you because you were burned by that terrible quiz question, right? Right? Remember? I brought my clock. Remember this? 5 primes on the front, 3 primes on the back. Person walks up to the back to make the bond, okay? So 3' hydroxyl is the right one. This was a popular answer, more so in A than in your class. But remember that when you cut a piece of DNA with restriction enzymes, and the 2 ends anneal, a hydrogen bond, did that take an enzyme for them to anneal? No. They form hydrogen bonds just because of their biophysical properties. They don't need an enzyme for that. DNA polymerase, its job is to make a phosphodiester covalent bonds. Okay, and they're phosphodiester, not ester, that's why that one's wrong. And, yeah. Okay. Try this. Okay, once again, I'm happy. It looks like you guys did review some of the material and you know your germ layers. So that's good. Yay! Okay, so C is the correct one. You know that muscle comes from mesoderm. So that was the right answer. Very good. Somebody have a question? No, I didn't hear questions? Okay. How about this one? Nice and short. All right, I think you guys did well on this one too. I'm building your confidence for Wednesday, so you better still study. Okay, A is right. So I especially wanted to include this one because at some point in some class I said something backwards. I said diamond when I meant kinesin, and I want to make sure we have this clear. So it is kinesin, which is a plus end directed motor. So the minus end of microtubules is anchored near the centrioles in the microtubule organizing center. The plus end is a rapidly growing end, and that's out in the periphery. So the neuronal cell body sending neurotransmitter out to the synaptic terminal is going to use kinesin to move it along microtubules. So that is the right answer. This is a nice one because it has a figure in it, and a lot of times you guys get worried when there's a figure and don't do as well, but it's not that bad. So give it a try. And voltage-gated channels are closed at the resting potential. So 50 milliseconds is at the resting potential. 400 milliseconds is back at that resting potential. So the right answer is 200. Yes? Oh, heavens. The sodium voltage gated channels be closed here and here. So if you're asking would the sodium channel be open here, I don't know. So this is a cardiac muscle zone. I don't want to tell you the wrong answer. But if we were looking at an action potential in a nerve, the sodium channels would be closed during that falling phase. So in this particular cell type, I don't want to say the wrong thing. It's a good question. Anybody else? Okay, let's see what's next. Aha! Okay, so we're going to try a couple short answers. Why don't you take a stab at this first? You guys feeling stuck? Yes? All right. I'll give you a little help. This was on one of the slides. Hopefully that helps. You can talk to your neighbor if you want to, if that'll help you. Candy, I forgot today. I should have done that. Does anybody have the right answer? What do they think? Yes. Yeah, sure. 5% is it? Good. Okay, very good. Congratulations, 5%. So this seems like it's hard, but it's not. And I think next year I'll probably change how I presented this a little bit because it shouldn't have been as hard as it was. So what you're looking for here is the recombination frequency. So we need to identify which offspring are recombinant. That's the key. So you have one parent is a gray body with red eyes. The other parent is a black body with purple eyes. So let's look for those parental genotypes. Gray body, red eyes, that's one. We have 362 of that parental phenotype. And then the other is black body, purple eyes. We have 398 of that parental phenotype. So that means these guys are the recombinants. Okay? So how many recombinants did we get? 18 plus 22 is 40. Okay? And then the number of total offspring, if you add all of these guys up, it's 800. So if we give you math to do on the exam, you're not bringing a calculator. So it'll be pretty simple stuff like this. So when you have 40 over 800, you can reduce that to 1 over 20, so it's 5%. Any questions, or is that okay? Yes. Am I going to give you this equation on the exam? No. I just gave you the equation here. We went through the problem in class. And so now I do expect you to be able to calculate a recombination frequency given this sort of information. So that's a good question. I want to make sure everybody knows that. Any other questions? Okay, let's try another one. This is a pedigree problem. So go ahead and try and figure out the inheritance pattern and explain. This is a short answer. So your ability to explain your answer is what gives you the points. So make sure you do a good job. And if you want to talk to your neighbors, that's fine with me. Okay, we have another brave volunteer. Tell me what inheritance pattern. Okay, I'm only going to say the second part. So he's saying it's recessive. Not sure about the first part yet. What's the next question? So what tells you, you may know that this is the next question. So what tells you that this is recessive?
Because even the 5 and 6, when they don't express it, they had a child that did express it, so they must have both been carriers.
That is absolutely right. So your big clue for a recessive inheritance pattern is affected individuals with unaffected parents. Okay, now crazy things can happen when things are sex-linked. So, you look at it and say, aha, this person is sick and their parents weren't recessive. You go through and make sure it's okay. But recessive is going to be a very good choice here. So what about this? So if you look at this, could this be sex-linked? Oh, that was fast. So you guys are so smart. Somebody, not you, somebody else, tell me why. Yes? Oh, yes, very good. So number 4 is a giveaway. That was fast. That's good. So if this was X-linked recessive, because we just said it's recessive, then this man would have had to get the Y from dad, and it would have had to get one of these guys from mom. Are there any other people that tell you this is impossible? Nine? Yep. 9 and 7 are both kind of in the same situation here. So if it's recessive, then these women had to get an X from dad that was mutant, and both of the dads are not affected. Okay, so the answer is X is recessive, no. So because of 9 and 7, and in addition number 4, all of them are good answers. So if you had to explain, well, why not? Any of those would be okay. Yes? Sure. Yeah. So the question was, if you're designating a mutant X chromosome, could you put X with a little n next to it? Yes. I mean, if you really want to be super safe, because I don't grade the short answer, I just tell them how to grade it. If you want to avoid hassle later, then you can just write XM equals mutant X chromosome. And then you're totally safe. But I would accept that guess. Okay, could it be X-linked dominant? No, we already said it's not dominant, but 7 also tells you it can't be dominant because the parents are not there. And no, it can't be Y-linked because there are females who are sick and are affected, and females don't have Y chromosomes. What about this? What is the genotype of number 4? You guys just told me this inheritance pattern is autosomal recessive. So what was the genotype of 4? Somebody in the middle. What is number 4? No takers? What's number 4? What's their genotype? Oh, in the back, yes. Heterozygous, yes. So why? Yes, I like your answer, and I'll repeat it. So I also use number 4 in my explanation, because if this is autosomal recessive, the only thing mom had to give was a recessive mutant allele. So they had to get a little a for mom, but yet they're not affected, so they had to get a big A from that. So the answer is heterozygous. Okay, this is to show you another kind of thing we might expect you to do on an exam. There's a simple drawing here. This curved line represents a chick limb bud, and the arrow points towards the anterior. So could you draw the ZPA and the AER on here? Do you remember what those terms mean? Don't just learn the abbreviations, know what they mean. So go ahead and you can scribble it down if you want. All right, so on these kinds of questions, following directions will be key. So if we say draw a circle, and that's where the ZPA would be. If we say write an X. Okay, don't do something different. Follow the directions is key. So that would be the way you would answer a question like this. Okay, how about this one? Well, my early answers were very correct, and my later answers were not so correct. So let's do the talk to your buddy thing again. Talk to your friend, and then re-vote. So this is what we have. Not sure why is a bicoid protein an inducer? You guys remember that was pretty much the only example of a cytoplasmic determinant. Okay, so inducers are things that tell other cells to do things. So bicoid, bicoid, however you want to say it, is not an inducer. It was given as a cytoplasmic determinant. Notochord. What does notochord induce? Neural tube formation. Neurulation, right. And notochord is what kind of germ layer? Mesoderm, whatever you want to say. Yes? Does the notochord become your backbone? That was one of the quiz questions, right? So I'm not a developmental biologist. It could, because your skeletal system, your vertebra, would be made of mesoderm, because it's skeletal. So I think so, is the answer. Something there. Some of the TAs is a developmental biologist will straighten me out later. Yes? Yeah, that's right. Okay, so the notochord is the primitive structure that disappears. It's all coming back. I had embryology a long time ago, but I do think that's right. So it kind of fades away. But I don't know if it like disappears as a structure and the cells go off to be somites or what. I don't think the cells all die. Those are good questions. You guys want me to do some more? Yes? Okay, you remember this, right? What's the answer? D. Good. What's the answer? D. Everybody hear D? 4 genomes copied. How many different alleles did I just circle? I heard someone say, I don't know. That's right. Because it could be the same allele on both of the homologous chromosomes. So it's either 1 or 2. Okay. I don't like that one or that one. Let's try this one, because this is a really important concept. We'll close out on this one. Well, you guys are doing much better than you did the first time when you saw this in class, so that's very good. The answer is C. Not everybody was on the page of this one, so study your notes. Epistasis, genes at other loci. And when you cross the purple and the white flower,